The overall goal of this procedure is to monitor changes in mitochondrial calcium concentration in live cells. This is accomplished by first transfecting cells with the calcium indicator protein ratiometric pericam, which is targeted to the mitochondrial matrix. 1 or 2 days following transfection, the fluorescence microscope and software is set up for live cell imaging. Images of ratiometric pericam expressing cells are then acquired upon addition of a calcium mobilizing agonist. The final step of the procedure is quantitative analysis of the acquired images. Ultimately, results can be obtained that show dynamic changes in mitochondrial calcium concentration through fluorescence microscopy of live cells expressing a ratiometric calcium indicator protein.
The main advantage of this technique over existing methods, such as measuring calcium levels using synthetic calcium indicators, is that ratiometric pericam is genetically encoded and thus can be targeted to mitochondria or any other organelle of interest.
Hello, I'm Dr. Askar Akimzhanov, a postdoc in Dr. Benning's laboratory, and I will be demonstrating the procedure today.
To begin the protocol, plate HeLa cells the night before transfection on sterile glass cover slips placed in a standard 6-well culture plate at a density that will be approximately 70% confluent for transfection the following day. The next day, prepare DNA lipofectamine 2000 complexes per the manufacturer's instructions with 4 micrograms of ratiometric pericam mitochondrial expression vector and 10 microliters of lipofectamine 2000 diluted in 0.5 milliliters of Optimem for each well to be transfected. Next, replace the DMEM FBS HeLa culture media in each well with 1.5 milliliters of the same media without antibiotics. Add the lipofectamine solution to the DNA solution and mix gently. After the complexes have formed, add the DNA lipofectamine solution dropwise to each well to be transfected. Mix by gently rocking the plate and incubate for 4 hours in a cell culture incubator at 37 degrees Celsius, 5% carbon dioxide. Following the incubation, replace the media with DMEM FBS containing antibiotics. Allow the cells to express ratiometric pericam for 1 to 2 days before imaging. Using forceps, gently place a cover slip with HeLa cells into an appropriate imaging chamber with imaging solution. Mount the imaging chamber in an inverted microscope stage. Next, use a 40x or higher oil immersion objective and a wavelength of 380 nanometers to find an area of interest containing 1 or more cells expressing ratiometric pericam. 380 nanometers is used here to identify the cells, as ratiometric pericam is less resistant to photobleaching at this wavelength. Once an area of interest has been identified, set up the software for dual excitation at 495 and 380 nanometers. Next, acquire images sequentially by alternatively exciting at 495 and 380 nanometers. Acquire images of baseline calcium levels for at least 30 seconds before application of agonist. Then apply the calcium mobilizing agonist via a perfusion apparatus, noting the addition time for each agonist. Exposure times and acquisition intervals should be optimized to prevent photobleaching while still allowing sufficient temporal resolution. Once the experiment is complete, images can be analyzed offline. To begin analysis, select a region of interest within an empty area of the field to subtract background fluorescence if necessary. Next, export the 495-380 ratio measurements from the region of interest to Excel or similar graphing software. This panel of images shows the subcellular localization of ratiometric pericam in mitochondria. On the left is a live cell image of HeLa cells expressing ratiometric pericam. In the middle is the fluorescent staining of the mitochondrion selective dye Mitotracker Red CMX-ROS, and finally on the right, the merged image. Shown here is a series of pseudocolor 495-380 nanometer ratio images of 4 HeLa cells expressing ratiometric paracam in the mitochondria treated with 10 micromolar ATP. And the quantification of changes in mitochondrial calcium levels of those previous 4 cells is shown here. In this image of a HeLa cell expressing ratiometric paracam in the mitochondria, heterogeneity of the calcium response in individual mitochondria, as well as significant fragmentation of mitochondria, is evident by 60 minutes of 0.5 micromolar staurosporin treatment. Some mitochondria have oscillatory increases in calcium, shown with the white arrow, whereas others do not show significant changes in calcium level, shown with a yellow arrow. This graph reveals the change in global mitochondrial calcium levels in a single HeLa cell after induction of apoptosis for 120 minutes with 0.5 micromolar staurosporin.
After watching this video, you should have a good understanding of how to measure dynamic changes in mitochondrial calcium levels in response to various stimuli using calcium indicator protein, ratiometric paracam, which is selectively targeted to the mitochondrial matrix.